Hey guys, let's talk about enzyme purification. Usually enzymes are associated with the term metabolism. Now metabolism is speeding up and slowing down the inner processes of the body. Now that's exactly what enzymes do. Enzymes speed up the reaction from this graph. This uncatalyzed becomes catalyzed when it's added, lowers the delta G, lowers the activation energy, and allows us to proceed from reactants to products much faster or in unnormal situations. So we proceed to our first step. We get a piece of tissue. Not a tissue box, but simply we have here is a heart. And in the heart, when you grab a tissue, a common procedure is to look for the cardiac enzymes. And if the enzymes are elevated, if there's more amounts of it, you have a suspected MI or a possible heart attack. Now that is important. So to proceed, once we obtain the tissue, we go to homogenization. And homogenization is basically putting our tissue into a blender or some type of machine that grinds up the cells, opening the cells. And we usually do this in something called a buffer solution. A buffer solution resists pH changes. Now it also helps with reducing oxidation and heavy metal ions, which could destroy the enzyme. Now, enzymes are also referred to as proteins, and that's why when you lyse up a cell, you have something called proteases, which is something that is also released that degrades protein, destroys our enzyme. So we want to do this in a 37-degree Celsius cold room or ice room or whatever we can get, and the biological enzymes or proteins that we want are still functioning, but the proteases are not because they're temperature dependent. We move on to the next step, which is sonication, which is just putting it in a long tube, and it uses ultrasound to create pores in the cell, allowing us to further grab the DNA or what we want inside the cell. We move on to our next step, which is centrifugation, which is very familiar to a lot of biologists, chemists, etc. Now, centrifugation is basically placing two tubes of equal amounts of volume in opposite sides of the centrifuge. And the centrifuge will spin at massive amounts of speed. 600 gravity, 20,000 gravity, 100,000 gravity. If you multiply gravity times this, you will get the RPMs, which is revolutions per minute. Now, inside the tube, you have a mixture of different things. I don't even know what's in there. Now, once you do that, you centrifuge it, you have a separation between supernatant and precipitate. Or dense materials tend to go towards the bottom and less dense will go to the top. And at different speeds, you're going to get different material, for example, the mitochondria in the pellet at 20,000 Gs. So moving to the next step, once you have it separated, you're going to want to proceed with salting out. Using ammonium sulfate, a very polar compound, this polar compound will bond with water and will prevent the protein or enzyme from bonding to water, allowing it to decrease in stability and then so forth, allowing it to bond to itself, which you will see some form of clumping. So the next step is dialysis. And dialysis is separating large molecule proteins from smaller molecules, such as salts and dyes. Now, you put your protein or enzyme of interest inside this semi-permeable bag. And the semi-permeable bag is made up of things like snakeskin or anything it could be. And it's clamped. Now, inside this beaker, you have a spinner spinning, and you have dialysis buffer solution. And when you spin it, the diffusion from high concentration inside seeps out to low concentration. And because it's a semi-permeable bag based on size, only the small molecules will leave, allowing some type of purification for your enzyme. Usually done twice. And then you move on to your assay, and you do your spectrophotometer to look for absorbance. At this point, comes the calculations where you do activity. And then you have your relative activity, which you can calculate. And then specific and fold purification are done with the Bradford.